Good afternoon, children. Good afternoon to everybody. Today, we are going to discuss about DNA fingerprinting. The topic is DNA fingerprinting. It is one of the last topic in molecular genetics. So, this DNA fingerprinting is first developed in some book they given the inventor. So, it is developed. So, the DNA fingerprinting is developed by the Alice Jeffreys. The word Alice Jeffreys. He only developed, that means he was developed the DNA fingerprinting in 1985. Some book they given the 1984. So you correct it in your book 1985. The DNA fingerprinting was first developed by Alice Jeffrey in 1985. This DNA fingerprinting was first discovered, that means invented or developed by the Alice Jeffrey. So he was called father of DNA fingerprinting. Father of DNA fingerprinting. Who was called the father of DNA fingerprinting? Alice Jeffrey was called father of the DNA fingerprinting. He did this experiment in the Leicester University, that is UK. He did the experiment in the Leicester University. Okay, next, Dr. V.K. Kashyap. Dr. V.K. Kashyap and Dr. Lord G. Singh. These two names didn't given in your book. So write in the book itself. Dr. V.K. Kashyap and Dr. Lord G. Singh started the DNA fingerprinting in India. DNA fingerprinting in India. It is one of the neat questions. Write in your book. Neat question. Who was the or who discovered DNA fingerprinting in India? Dr. V.K. Kashyap and Dr. Lord G. Singh. So they did the experiment in the Center for the Cell Molecular Biology. That is a CCMB. CCMB means Center for Cell and Molecular Biology. This institute at Hyderabad. There only the Kashyap and Dr. Lord G. Singh started the DNA fingerprinting. Okay, so the Dr. Lord G. Singh was called the father of Indian DNA fingerprinting. Normally, the world-wise we are asking question, who was the father of DNA fingerprinting? Alice Jeffrey. That is a worldwide. In our India, we are taking this. Who was the father of the Indian DNA fingerprinting? Means Dr. Lord G. Singh. What is the name? Dr. Lord G. Singh. The DNA fingerprinting is otherwise called. The DNA fingerprinting is otherwise called DNA typing or DNA profiling. DNA fingerprinting is otherwise called DNA typing or DNA profiling. So first we want to know about what is DNA fingerprinting. DNA fingerprinting. The DNA fingerprinting means laboratory technique. It is one of the laboratory technique. One of the laboratory technique. Right there itself. What is the meaning of that is a DNA fingerprinting? DNA fingerprinting is a laboratory technique used to establish link between biological evidence. Link between biological evidence. Link between biological evidence and a suspect in a clinical investigation. Clinical investigation. It is one of the definition for the DNA fingerprinting. So once again, I am saying about the definition for the DNA fingerprinting. The DNA fingerprinting is a laboratory technique used to establish a link between biological evidence and a suspect in a that is a clinical or criminal we can take. That is not a clinical. We can take the criminal investigation. So it is a very important usage in the criminal department. So with the help of the DNA fingerprinting, we can identify or we can find out the criminal things. That is in the criminal department we are using the DNA fingerprinting. Okay, this DNA fingerprinting is represented with the blueprint. It is representing blueprint of the human genome method. Human genome method. You know what is genome? The DNA and RNA, the nucleic acid structures we are talking the same about the genome method. So the DNA fingerprinting represents the blueprint of the human genome method. Blueprint of the human genome method. The 2 points you want to keep in your mind. Fix it. That is what is DNA fingerprinting? DNA fingerprinting is a laboratory technique. It is used to establish a link between biological evidence as well as the criminal that is a investigation. Criminal investigation. So this DNA fingerprinting represents the blueprint. It is representing the blueprint. Blueprint of the what? Blueprint of the human genome method. Blueprint of the human genome method. Okay. So with the help of this DNA fingerprinting, we can compare the different kinds of the human genome methods. Okay. So it is determining the nucleotide sequence. What is the nucleotide sequence? You already in the same lesson, the first class, you studied about the structure of the DNA as well as the structure of the RNA. In that one, the structure is made up of the 3 parts. One is a sugar, another one is a nitrogenous phosphate, another one is a phosphate group. Based on the nitrogenous group, so that the DNA contains the adenine, guanine, cytosine, and thymine. So RNA contains adenine, guanine, cytosine, and uracil. Instead of the thymine, uracil is present in the RNA. These are the nucleotides. When one glucose, that means one sugar molecules, combine with the one nitrogenous bases, that will make the nucleoside. One nucleoside combined with the phosphate group, that will make the nucleotide. You already studied what is nucleoside and what is nucleotide. One sugar molecule plus one nitrogen bases, it will give the nucleoside. That structure is known as what? Nucleoside. One nucleoside plus one phosphate group will give the one form that is called the nucleotide. Do you understand what is nucleotide now? So, to determine the nucleotide sequence, that also especially in the DNA area. You already know the DNA structure. That is a double helix molecule or double helix thread-like structure. So, in certain area, these are in the nucleotide, which is unique in each individual. Each and every individual has a different type. That means separate type of the nucleotide. The nucleotide is not the same in all the individuals. So, somewhat different is that. It is an ideal way to distinguish an individual from other people. In this way only, we can identify the differences. We can see the differences in between the people. Which one basis? The basis of the nucleotide sequence. Where the nucleotide sequence is present, it is present in the DNA. So that means what? DNA is not same in all the people. It is not the same type of the nucleotide in all the people. So we can't alter this nucleotide also. We can't alter the nucleotide. This is one of the nucleotides. If the nucleotide can alter, the nucleotide cannot alter by any method. That is what we are having in our from the birth. The same type of the nucleotide is present in the body. There are how many chromosomes we are taking? 23 pairs of the chromosomes are present in the human body. 23 pairs means how many chromosomes? 46 chromosomes are present in the human body. Okay. So out of this 23 pairs of chromosomes, and the number of the genes, that is one of the number of the genes. The genes are present like a pairs. So totally how many pairs of the genes are present in our body we are taking is 1.5 million of the genes. Need to see. So how many pairs of the genes present in the human body? 1.5 million of the pairs of genes present in the human body. Where it is present, all the genes are present in the chromosome. So in the chromosome, where? In the DNA. The genes are the segment of the DNA. That means what? We have the chromosome. The inside of the chromosome, that is having the DNA. So the DNA consists of the number of the genes. So the genes are the segment of the DNA. The genes are the segment of the DNA, which is differing from the sequence of that means one nucleotide to the another nucleotide. Not all the segments of the DNA code for proteins. All the segments of DNA code for proteins. Not all the segments are the protein codes. Some DNA segments are revolutionary function. That means it will make the regulatory function. Some DNA are making the regulatory function and others are making the intervention sequence. intervention sequence. That is called the introns. Introns, exons, like that you already studied. Introns and exons. These are present in the DNA. So before dropping protein synthesis, in that one you studied about the introns. Introns means what? Intervention sequence. So this where the intervention sequence are present, it is present in the DNA sequence. Where the DNA segment is present, it is present in the chromosome. So some DNA segments have regulatory function, and some are other inventory, that is a intervention sequences. Still others are repeated DNA. Some DNA are doing regulatory function. Some DNA are like a intervention sequence. That is like a intron and the extron. So some are the repeated DNA sequence. Some are the repeated DNA sequence. You understand? This one is about the introduction about the DNA fingerprinting. So in this one, you want to know about who was the father of the DNA fingerprinting. Alice Jeffrey was the father of the DNA fingerprinting. Okay. Who was the DNA fingerprinting in India? Who was the father of DNA fingerprinting in India? So Dr. Lorgisi. Okay, that's two things you want to remember. Next, what is DNA fingerprinting? DNA fingerprinting is a laboratory technique used to establish a link between biological evidences and a suspect in a criminal investigation. So this DNA fingerprinting represents the blueprint of the human genetic network or human genome network. Genome means also what? Genetic. Human genetic network. The segments, the DNA segments are a regulatory function and the intervention sequence as well as the repeated DNA sequence. This is about the introduction about the DNA fingerprint. Next. Next one. DNA fingerprinting principles. That means principles of DNA fingerprinting. Principles of the DNA fingerprinting. So, first we are taking the principle of the DNA fingerprinting that each and every individual, that means each and every individual in the human being, each and every individuals of the human being has have a 0.1% to 100%, that is a totally nucleoside basis. That means individuality, each and every individual as in a human being has a 0.1% to 100% of the nucleotide, that is DNA particles in the chromosome, except the monozygotic twins. So what is monozygotic twins? The monozygotic twins are known as the identical twins. Monozygotic twins are called what? Identical twins. In the second lesson, you already studied about the different type of the twins. Identical twins. Monozygotic twins are known as the identical twins. These identical, why it is called the identical twins? These are producing when a single fertilized eggs split into two first cleavage, two individual or two gametes, or two otherwise called the two during the first cleavage. See, they are produced single fertilized egg split, completely split into two daughter cells. That means two daughter cells will produce separate cells. Each cells will develop as a individual. So these are the same sex in the identical twins. Do you understand? Identical twins only, otherwise known as what? Monozygotic twins. In this monozygotic twins are produced, so that is a single egg, that means single fertilized egg split into two daughter cells. So in the first cleavage itself. So there are the same sex as well as look alike. This one looks like alike. Alike means like a same. Looks like a same. As well as share the same genes. It is very important one. Share the same gene. That means both individuals have the same genes. That is one of the characteristics of the identical twins. So second type one is known as the dizygotic twins. Second type, dizygotic twins. These dizygotic twins are otherwise called the fraternal twins. It is called what? Fraternal twins. Dizygotic twins are otherwise called the fraternal. In these twins, these are produced when two separate eggs are fertilized by the sperm. Separate eggs are fertilized by the two separate sperms. Two eggs are fertilized by two separate sperm. These are same sex. Sometimes it is a same sex, and sometimes it will change as a different sex. That means non-identical. It is non-identical. Do you understand? This type of the twins are known as the dizygotic twins, or otherwise called the frontal. So in the monozygotic twins, we can't see that any genetic differences. All the different, that is gene conditions are same in the individual. That means daughter sex. So only it is individuality is present in each and every human beings. That means the chromosomes, that means nucleotide sequence are not same in all the human beings. It will differ from one person to another person, except monozygotic twins, because the monozygotic twins are formed from only one fertilized egg. So only the genes are shared there. So the monozygotic twins are, that means identical twins are have the same kind of the genes. That is one of the important. But the dizygotic twins are have the what different type of the genes, because the nucleotide sequence are different from the one organism to the another organism. The human genome, numerous small non-coding sequence. Just a small non-coding sequence. That all the things are veritable ones. Veritable means what? It is transferring from one generation to the another generation. So the non-coding genes are gone. Some genes are not carrying the code. That means already studied phenylalanine, alanine, the amino acids are carrying the like a codes. You already studied like AU and AUG, like that you studied about the triplet code. These triplet codes are carried. Some genes are not carrying the any codes. That is called non-coding genes. The non-coding DNA or otherwise called the junk DNA. That is called the junk DNA, or otherwise known as inactive DNA. The inactive DNA means it is not doing any function. You know already all the nucleotides are present in the double helix model. Like that is the DNA structure is there is having the nucleotides adenine, thymine, guanine, cytosine, adenine, thymine. So like that there is having the code. There is a nucleoside as well as a nucleotide like that. Okay. So these are all the things, these codes are. These codes are in some places it is inactive in condition. That region is known as the junk DNA region, or otherwise called the inactive DNA. That is not doing any other function. So totally 0.1% to 100%. That means 99.1% of the nucleotides are same for every individual. More or less same in each and every human being. All the human beings are in the same type of the nucleotide sequences. That means how many percentage? 99.29% also we can take. Only except 0.1 differences. 0.1 difference occur near the telomere or centromere or chromosomes and heterochromatic area. In the DNA, we can see the telomere, centromere. You already studied about the telomere. The presence of the centrioles, that where it is present in the DNA, according to that one, it will get the telomere, centromere, like that name. So in that region, so the 0.1% differences we can see. So totally 99.9% of the nucleotides are same for each and every individual. Only 1%, that means also 0.1% is different from one person to another person. That difference we can see in the telomere, centromere, Y chromosome and heterochromatic area. So based on the density gradient, the density gradient we can do with the help of the centrifugation method. We can do the centrifugation method. So based on the density gradient, the DNA are divided into two types. Based on the density gradient, we can divide the DNA into two. So one is called the bulk DNA. One type is known as what? Bulk DNA. Bulk DNA. Another one type we can take, that is here, satellite DNA. Another one type is known as what? Satellite DNA. You understand? The DNA is classified based on the density gradient into two types. One is the bulk DNA, another one is the satellite DNA. On the basis of adenine, thymine, guanine, cytosine, I already told the DNA nitrogenous compounds are adenine, guanine, cytosine, thymine, adenine, guanine, cytosine, thymine. These are the DNA nitrogenous base. DNA nitrogenous base. Nitrogenous base. Based on the nitrogenous bases, as well as the length of the sequence, number of the time sequence repeats. Number of the time the sequence repeats. I already told. So the DNA we are taking is like that. We are taking the DNA. We are taking like that DNA, adenine, thymine, cytosine, guanine. Once again adenine, thymine. So that is called the repetition. That is known as what? Repetition. So based on that repetition, the number of the time the sequence repeats, the satellite DNA is further divided. The satellite DNA is further divided. The satellite DNA is further divided. Further divided into two types. The satellite DNA is further divided into two types. So one is called the mini satellite. One is known as the mini satellite. One is known as the mini satellite. So the second one is known as the microsatellite. Second one is known as microsatellite. So you want to know about this classification. Okay, the DNA is based on the nitrogenous bases. Nitrogenous bases only we are saying that adenine, thymine, cytosine, and guanine. As well as the length of the sequence, as well as the time of the repetition of the adenine, guanine, thymine, cytosine. So the satellites are divided into two types. Mini satellite as well as the microsatellite. Okay. Now the satellite of DNA, that is a satellite DNA. You already know which one is the satellite DNA. The DNA is classified into two types based on what? Density gradient. Based on the density gradient. Normally the DNA is classified based on density gradient into two types. One is bulk DNA, another one is the satellite DNA. Now the satellite DNA only further divided based on the nitrogenous bases and the length of the sequence and number of the time the sequence is repetition. So it is classified into two types, mini satellite and microsatellite. Now the satellite DNA form a large portion of the human genome. The satellite DNA forms the satellite DNA forms one of the large portion of the human genome. This sequence show SH word will show high degree of the polymorphism. High degree of the polymorphism is what is polymorphism? The many characters are present there. That is called the high polymorphism and forms the base of the DNA fingerprinting. Which one is formed the base of the DNA fingerprinting? The satellite DNA, not DNA. The satellite DNA only forming making the basis of the DNA fingerprint. Do you understand? That is one of the lead questions. Which DNA forms the base of DNA fingerprinting? Whether it is a DNA, RNA, or satellite DNA, or bulk DNA. So, which option you want to select? Satellite DNA. So, the satellite DNA only forms the base of DNA fingerprinting. Okay, I already informed about the polymorphism. The polymorphism only shows the variation. We know what is variation. We can see the difference from the one organism to the another organism. That is called the variation. Sometimes the mutation can also take place. Where in non-functional DNA part, I already told you. So all the parts are helpful for doing the function, except some part. That means we can take the 0.1% like that. So in that part, only the non-functional part only we are saying about what junk DNA, or otherwise called the inactive DNA. In that region, suppose any mutation takes place, it will do the what? Somewhat, it will show the somewhat variation. Do you understand? All the part of the DNA will give the full function, except some region of the DNA. That region only known as, or that area only known as the junk DNA, or otherwise called the inactive DNA. So in that region, sometimes it will make the showing the mutation. So for that mutation means what? Sudden change. Mutation means sudden change. Because of the sudden change, some variation takes place in some area. That which area? Non-functional area. So what is non-functional area? The non-functional area only known as the inactive area of the DNA. Inactive area of the DNA. So inactive area is otherwise called what? Zunk area of the DNA. Zunk area of the DNA. Do you understand? In DNA fingerprinting, short repetitive nucleotide sequence are specific for a person. I already told you. The nucleotide is same for all the human beings. In some area, that is 0.1% only, the nucleotide variation is occurring from one person to the another person. For example, person A we are taking, person B we are taking. So 99.19% is the nucleotide is same for both person A as well as, for example. So there is only some differences. That is the 0.1% differences in between the nucleotide. With the help of these differences only, we can find out the differences in between the one person to another person. That is a genome condition. So these nucleotide sequences are called the variable number tandem repeats. That is called what? VNTR. VNTR means what? What is VNTR? VNTR means variable number tandem repeats. Variable number tandem repeats. These variable number tandem repeats are occurring from one person to another person. We can take the differences with the help of VNTR. Because 99.9% same genome, but only 0.1% only different genome. That sequence part only known as the variable number tandem repeats. See, in this diagram you can see an example. Chromosome 7 is there. Chromosome 2 is given here. Chromosome 16 is given here. See, in the chromosome 7, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. 11 segments are present. In the chromosome 7, another one pair is the 2 chromosomes are. So, out of chromosome 7 pair, the first chromosome, this one second chromosome. The second chromosome contains 3, 4, 5, 6, 7, 8, 9. Only the 9 segments. Like that, see in the chromosome 2. First pair, the outer pair, first chromosome. First chromosome has a 8 segment. Second chromosome has a 6 segment. In the 16th chromosome, see, first chromosome has a 5 segment. And second chromosome has a 2 segment. Okay, this one we are applying in the, or putting in the gel. That is like this pad-like structure is present. Then we are representing these chromosome segments there. That means in the nucleus there is a chromosome. That chromosome is we are making a fragment because always the chromosome is thread-like structure. We are making the chromosome as a fragment. So with the help of the endonuclease, restriction endonuclease. You already studied, you know very well about the function of the restriction endonuclease. With the help of the restriction endonuclease, we are making the small fragment in the DNA part. So the DNA is representing on the surface of the another one region, that is a gel-like plate. So that is we are plotting this DNA. Now see the first segment, 9, 11. So we are arranging. That is a descending order, 12, 11, 10, 9, 10. So we are arranging this 11. 11 is the highest one. So we are arranging 11. And the second one, that is a 9. So we are arranging the 9 is the red color code. You remember, the 11 also red color code, 9 also red color. Next 8. So that is a green. Next 6 green. Next one see is the 5 blue color. And 2. So in this one, the same color is repetition is takes place in the 2 types. That type of the repetitive condition or repetitive nucleotide only, we can see that with the nucleus. That is VNTR nucleus. That is a genome or VNTR DNA. That is called what? VNTR DNA. So we are plotting like this on the surface of the gel. The gel plate is separating and making the amplification, and we can find out that which one is repetition is takes place, which color is repetition is takes place. So from that one, we can understand which DNA is making the repetition. That part or that region or that area only known as the variable number tandem repeats. Variable number tandem repeats. So the VNTRs of the 2% generally show the variations and are useful for the genetic markers. That is useful for the genetic markers. DNA isolated from the where we will take the DNA. We can take the DNA from the cells. So from which cells we are taking the DNA? From blood, hair, skin cells, and other genetic evidence. That means sperm, womb. From that also we can take the DNA for making the test. That is a crime can be compared to the DNA. The crime branches are using this method only and find out in the our criminal persons with the help of the DNA test. That test only known as VNTR test. We understand? So the VNTR test is helpful. One maximum it is helpful for the number of the field we are using this one. So the next episode we will see about the application of this DNA fingerprinting. But DNA fingerprinting they are using the what method VNTR patterns. What is meant by VNTR patterns? That is a nucleotide is making the repetition in a person's body. So with the help of that repetition, we can find out that is any the defect is takes place means we can find out with the help of the VNTR patterns. These VNTR patterns are useful in establishing identity of the homicide victims. We can identify the victims. So any drawback we can identify with the help of the VNTR method. That is what is meant by VNTR? Variable number tandem repeats. Do you understand? So next, how we can take or how we can find out this VNTR method? That means what are the steps we can make to making the DNA fingerprinting technique? So next one, we are going to discuss about the steps in DNA fingerprinting. The steps in the DNA fingerprinting in. So it is one of the 5 mark question in the theory part. So you want to draw like this diagram and you can explain the same point. That is very easy for you. So with the help of the diagram itself, you can explain these things. No need to read fully from the book. See, what are the steps are takes place in the DNA fingerprinting? Okay. So first, the steps in DNA fingerprinting technique. First steps, the DNA is extracted from the hair, saliva, sperm, blood sample. Anyone, not for all the things. Anyone we can take the DNA from this one of the things. That is from the hair or from the saliva or from the sperm or blood or samples. So here in the book they given the blood sample. So first we want to DNA is extracted from the blood. Next, DNA is extracting, this is a blood sample. From the blood sample, we are extracting the DNA. This is one. DNA is extracting from the blood sample. Okay, next, the DNA is making the broken. Broken is what we want to make the number of the pieces. So the DNA is cutted into fragments by the enzyme that is known as the restriction enzyme. That is called what? Restriction enzyme. Endonuclease. Restriction endonuclease. Restriction endonuclease. That enzyme is helpful for making the fragments. That means cutted the DNA into number of the pieces. The pieces only known as what? Fragments. The second point what? DNA broken with restriction endonuclease. What is the name of that enzyme? Restriction endonuclease. With the help of the restriction endonuclease enzyme, the DNA is cutted into fragments. Fragments means what? Number of the pieces. Next, the DNA pigments are separated into bands using the agarose cell. Very, very important one. All the DNA fragments are separated. First, the DNA looks like a thread-like structure. So, which is present in the blood sample. Only blood sample only we are taking. From the blood sample, the DNA is extracted. Then the extracted DNA is cutted the number of the fragments with the help of the restriction enzyme. Then the restriction in the DNA fragments are separated into bands like this. Separated into bands. So with the help of what? Agarose. That is a agarose jet. That is called what? Agarose jet. That is called agarose jet. That is during the electrophoresis. During the electrophoresis or by the electrophoresis. You understand? First step, blood sample. Second step, DNA is extracting from the blood cell. Third step, the DNA making are cut into fragments. Then fourth step, the DNA fragments are separated into bands. During that is the bands by the help of the agarose gel by electrophoresis. Next, these are making the VNTR separated and multiplied using the PCR poly chain reaction. The DNA band pattern in the gel is transferred. That is making what one of the VNTR test. I already informed about the VNTR test. Okay, with the help of the VNTR test, these are separate and multiplied using the PCR poly chain reaction. Okay, they split into a single stranded. So all the things are double stranded. I already informed DNA are double stranded. That are converted into or split into single stranded by using the alkaline chemical. We understand? First, we are taking the blood sample. From the blood sample, we are removing the DNA. That means extracting the DNA. So after the extraction of the DNA are making the number of the fragments with the help of the restriction enzyme. Then this DNA are separated into bands using the agarose gel by the electrophoresis. Then this DNA are split. That means double stranded DNA are split into single stranded DNA by using the alkaline chemical. Next, the DNA band pattern in the gel is transferred to the nylon membrane. Then it is transferred to the nylon. It is transferred to the nylon membrane. That technique is known as the Southern blotting. The technique is known as what? Southern blotting. Okay, the blotting is filtration, making the filtration or making the filter. That is called the blotting. So under the blotting has 4 types. There is a 4 type of the blotting. That is not given in the book. You make right value book itself. How many type of the blotting? 4 type of the blotting. Here given what type of the blotting? Southern blotting. The Southern blotting is detected only DNA transfer. That means Southern blotting is useful for transferring the DNA. Transferring the DNA. Which one? Southern blot method. So the second method is that is called the Northern blot. Another one method that is known as what? Northern blot. I already informed blotting means what? Blotting is making the what? Filter the things. Okay, so this blotting itself, we can write that itself. Blotting is one of the technique for analyzing the biological molecules. Analyzing the biological molecules. Blotting is what? One of the technique. It is one of the technique. It is useful for analyzing the biological molecules. Biological molecules. Do you understand? That is called the blotting. So this blotting only divided into 4 types. So here given the Southern blot. With the help of the Southern blotting, we can detect the DNA. Northern blotting, the second type is Northern blotting. It is useful for detect the RNA. This method is useful for the RNA detection. The third one is called the Western blot. Western blot. This Western blot method or blotting is helpful for the detecting the protein. Western blot. Next, Eastern blot. The Eastern blot is helpful for detecting the carbohydrate. Remember this one? It's one of the need question mark, not given in your book. So you write that. So blotting means what? Blotting is one of the techniques. With the help of the technique, we can analyze the biological molecules. So what are the biological molecules? DNA, RNA, protein, carbohydrate. These all the things are one of the biological molecules. These are detected by the help of the blotting technique. So Southern blotting is helpful for detect the DNA. Northern blot is helpful for detect the RNA. Western blot is helpful to detect the protein. And Eastern blot is helpful to detect the carbohydrate. Okay. Now the DNA is probe bands. So after we are transferring to the nylon sheet. So with the help of the blotting method, the is making the filtration. So next one. So the radioactive DNA probe is burned. Next we are preparing the DNA probe. The DNA probe binds to specific DNA and it is bind on the surface of the membrane. The probe is bind on the surface of the membrane. So which probe? DNA probe is bind to the specific DNA sequence on the membrane. The excess, some probe is bind on the DNA membrane. Other probe is washed off. Other probe is washed off. Do you understand? First blood sample, then get the extraction of the DNA. Then the DNA is cut into the fragments by the enzyme that is called the restriction endonuclease. So after that is clotting, that is the fragments are separated into bands by the electrophoresis method in a agarose cell. So then the DNA pattern is transferred to the myelin sheath. Then the radioactivity DNA is prepared. The radioactivity DNA is so sent to the this part, that is a DNA part. Some radioactivity probe is attached, the bind or attached on the surface of the membrane of the DNA. And the excess of the DNA probe is washed out. At this stage, the radioactivity probe is bound to the DNA pattern on the membrane. Some probe is bounded on the membrane of the DNA, and some of the probe is not bounded. So that are not bounded DNA are washed out. The X-ray plate is placed next to the membrane. Now we are keeping the X-rays. That is a DNA probe with bind to the DNA sequence of the membrane. Next, X of the DNA is that is a probe is washed out. Then at this stage, the radioactivity probe is bound to the DNA pattern on the membrane. This is next one. X-ray flame is up above X-ray. This X-ray flame is placed next to the membrane of the radioactivity pattern. So the detect the radioactivity pattern. Now the X-ray film is developed. Many, not male, many visible patterns. Many, it is the X-ray film is developed to, so they make visible. You can take that, make visible the pattern of the bands, which is known as the DNA fingerprint. This is something known as the DNA fingerprint. Do you understand? The first, we are taking the one sample. So whether in the diagram, we are taking the blood sample. From the blood sample, we are extracting the DNA. Then the DNA is cut into fragments by the enzyme that is known as the restriction enzyme, that is called the endonuclease. Restriction endonuclease. Restriction endonuclease. After that one, restriction endonuclease, that is DNA fragments are separated into bands using the one of the gel that is known as the agarose gel. So by the what method? Electrophoresis. Next, this DNA are separated and multiply. This one is separated as also multiplied by the PCR. That is not given in a book. You make it right there. So that is what the VNTR. You know already know what is VNTR. That is making the number of the repeated copies of takes place in the DNA. That is VNTR. Okay. So in this VNTR, so making the separating and multiplying using the PCR method. So next, split into single stranded DNA into a, that is single stranded DNA from the double stranded DNA using the alkaline chemical. With the help of the alkaline chemical, the double stranded DNA is split into single stranded DNA. The DNA pattern in the gel is transferred to the nylon membrane by a technique. That technique only known as the Southern blotting. The Southern blotting is helpful to transfer the DNA or to detect the DNA. But suppose RNA means we can use the northern. Suppose you want to find out the protein, we want to use the western blot. So next, carbohydrate means we want to use the eastern blot. But here we are going to find out DNA, so only we are using the Southern blotting method. After the Southern blotting, then the DNA probe is prepared. The DNA probe is bind to the specific DNA sequence membrane. The DNA probe is binds to the specific DNA sequence membrane. Okay. Okay. The DNA probe binds to the specific DNA sequence on the membrane. Next, X of the probe is washed out. Then at this stage, the radioactivity probe is bound to the DNA pattern of the membrane. The DNA pattern of the membrane is bound with the DC of the probe is bound with the DNA pattern of the membrane. Now we are keeping one of the X-ray film next to this membrane, next to the this membrane. That means for which membrane? That is a DNA pattern on the membrane to detect the radioactivity pattern. Now it is detecting the X-ray film developed to make visible pattern of the bonds. Visible pattern of the bonds. That pattern is only known as the DNA fingerprint. That pattern only known as the what? DNA fingerprint. The X-ray field is developed into developed into many visible the pattern. Many visible the pattern of burns. The visible pattern of the burns only known as the DNA fingerprinting. That only known as DNA fingerprinting. Like this only, the DNA fingerprinting technique takes place. These are the steps we are following to making the DNA fingerprinting. Okay, do you understand? Next, application. Next one, application. Next, you want to know about the applications. For this application of DNA, where we are using this day-to-day life. First one, we are using the forcenic analysis. Forcenic analysis. What is forcenic analysis? Criminal activities. So it is used in the identification of a person. Those who are involved in the criminal activities, we can find out the person with the help of the EATR method or otherwise the DNA fingerprinting. Forcenic analysis is the identification of the person. Okay, it is using the settling the paternity and maternity disputes. Suppose any paternity difference between the mothers or maternity, maternity, that is a parents were the child. Any disputes are there, means that also analyzing with the help of the DNA fingerprinting. That is settling paternity or maternity disputes. That means disputes means somewhat problems or takes place in the mother as well as the child. That we can easily clear with the help of the DNA fingerprinting. Suppose the mother is there, so the child also two childs are there. So we want to find out the child is belong to this particular mother or not. So the two mothers are also saying that the child is mine. So like that, any problem is coming in that time. So we can use the DNA fingerprinting method and we can find out. So which is the child is belongs to which parent, or the parents' baby is which one is the parents, that particular parent's child. So we can find out with the help of the DNA fingerprinting. Next one, it helps to determine the relationships for the immigration purpose. Immigration means they are moving from outside to inside. That is called the immigration. Moving from one place to the outer region means that is called the immigration. So they determining the relationship for the immigration purposes, we can identify with the help of the DNA fingerprinting. Next, it is helpful to analyzing the inherited diseases. That is, we can say the pedigree analysis. That is, we are saying the pedigree analysis. So with the help of the pedigree analysis, that means what is the DNA fingerprinting, we can analyzing the inherited diseases. What is inherited diseases? Can you give any example for inherited diseases? The disease which can be, that means for example, gonorrhea, syphilis, these are the inherited diseases. You already studied in the 4th class about the inherited diseases. The color blindness, okay, phenylketonuria. So these are the inherited diseases. These type of the diseases are transferring from one person to the piece, from mother to the children, mother to the child. So like that we can say the night blindness, color blindness. These are the inherited diseases. Color blindness. The 4th lesson you studied about the color blindness. Okay, phenylketonuria, night blindness. So like this, different type of the disease, that is we can say that is hemophilia. Okay, so this type of the inherited diseases are detected with the help of the DNA fingerprinting. Next one. Conservation of the biodiversity. Next one, it is helpful for the conservation of the biodiversity. That is a protection of the endangered animals. Extinct animals one, and the other one is endangered animals. Extinct already, that is what destroyed. Going to destroy, that is called the endangered species. We understand? Extinct organism means already destroyed. So there is no evidence, or no, there is no anyone, does not have only one species in the world. That is extinct. So endangered, going to extinct, that species we are protecting with the help of the DNA fingerprinting. That is protection of the endangered species by maintaining the DNA records. By maintaining the DNA records for identification of tissues of the dead endangered organisms. So we can produce or we can that is making the new organisms. So in that period, that is conservation of the wildlife also using the DNA fingerprinting method. Next one. Anthropological studies. Anthropological studies. It is useful in the determining the origin and migration of the human population and genetic diversities. Human population as well as the genetic diversities. That is known as the anthropological studies. So in the anthropological studies also we are using the DNA fingerprinting, that is origin and migration of the human population and the genetic diversities are takes place coming under the anthropological studies. These are the application of the DNA fingerprinting. This is also one of the 5-mark question in the theory. Write about the application of the DNA fingerprinting. First one, forensic science. That is a forensic analysis. Forensic analysis is used in the identification of the person involved in the criminal activities. Second, it is used for settling the paternity as well as the maternity disputes. Next, third one, it helps to determine the relationship of the immigration processes. Next, it helps to detect the inherited diseases by the pedigree analysis. Next, conservation of the wildlife. Protection, we are giving the protection. We are giving the protection to endangered species. We are giving the protection to endangered species by maintaining the DNA records. Next, anthropological studies. It is useful in determining the origin as well as the migration of human population and genetic diversity. What are the genetic variations are there in the one organism, one human being to the another human being. From some human beings came from the Africa. So after that is migrating from one region to the other region. Like that we studied in the evolution lesson. So that also we are getting the, without confusion, we are getting the clarification by the help of the DNA fingerprinting. You understand? So next we are going to see the multiple choice question. What is given in your book? That only we are seeing. So in the book itself, you mark the answer about the 5th lesson. About the 5th lesson. Do you understand? Now, 5th lesson, first question. In your book, exercise question. These are. So Hershey and Chase experiment with bacteriophage showed that. Sir, excuse me. Pasal பேசுறேன். So today now I'm going to discuss about the book exercise 14 onwards more. See, Hershey and Chase experiment with bacteriophage showed that which option is correct answer? Can you say? Can you hear my voice? Hershey and Chase experiment with bacteriophage showed that. Which answer is correct? Can you say the answer? Protein gets into the bacterial cell. DNA contains radioactive sulfur. DNA is the genetic material. Viruses undergo the transformation. Which answer is correct? Can you say?
DNA is the genetic material.
John, DNA is the genetic material. Okay. Second question is DNA and RNA are similar with respect to. Which the option is correct? DNA and RNA are similar with respect to. DNA, RNA are similar. Thymine as a nitrogenous base is the DNA. RNA we are taking is uracil. So that answer won't come. Okay. A single stranded edits shape. DNA double stranded, RNA single stranded. So that also similar only they ask the question. Can you say which one? The nucleotide containing sugar, nitrogen base and phosphate. I already told you. Nucleoside is formed by the help of the sugar molecule plus nitrogen base. That will form the nucleoside. Sugar plus nitrogen bases plus phosphate. that will form the nucleotide. So the nucleotide is common in the DNA as well as the RNA. So the nucleotide containing sugar, nitrogen bases and phosphate. That answer is correct. Next one. A messenger RNA molecule is produced by. Can you say the answer? Can you say the answer, ma? Messenger RNA molecule is produced by. Already this topic we have taken for you. Discuss. Replication, transcription, duplication, translation. Don't know.
Transcription.
Transcription. Only one student only saying the others. What about others? The total number of the nitrogen bases in human genome is established. How many zeros? How many millions?
3.1 billion.
3.1 million or billion?
Billion.
3.1 billion.
Okay. So next one. The same portion is the test portion for you next week. So only I am discussing this with 14 onwards now. Okay. So mark in your book. Already marked in the book, given the dark letters, but whether it is right or wrong, you want to verify that one now answers. Okay. E. coli cell grown on 15N medium, that is nitrogen medium, are transferred to the 14N medium and allowed to grow of 2 generations. DNA extracted from these cells is ultra centrifuged in a cesium chloride density gradient based on the density gradient. What density distribution of the DNA would you accept in this experiment? What are the density you are expecting? Can you say the answer? 1 row and 1 intermediate density. Now what I told, the DNA are classified under the DNA fingerprinting based on the density gradient. The DNA are classified like that I told. Next, what is the basis for the differences in the synthesis of the leading and the lagging strand of DNA molecule? Can you say the DNA polymerase can join new nucleotides only to the that is a 3 prime. This one to pronounce 3 prime. 3 prime end of the growing strand. Okay, next one. Which of the following is the correct sequence event with reference to the central dogma? Already thought that one central dogma about the central dogma. Can you say that one? Answer which one answer is correct?
Replication, transcription, translation.
Transcription and translation. Okay, next one. Which of the following statements about the DNA replication is not correct? I am asking the question, not correct. Which one of the following statement is about DNA replication is not correct? Can you see the answer? Not correct. Which of the following statement about DNA replication is not correct? Replication occurs as each base is paired with another exactly like it. That is not correct. Mark it in your book. Next one. Which of the following statement is not true about DNA replication in eukaryotes? Do you know what is eukaryotes? Can you say what is eukaryotes?
Multi-celled organisms.
The nucleus is cleared. That means well-developed nucleus means that is called the eukaryotes. Multi-celled nucleus. Well-developed nucleus is present means that is known as the eukaryotes. The nucleoid material is present means that is called the prokaryotes. The prokaryotes example bacteria. Eukaryotes example iron animals. That means well-developed nucleus is present. That is called the eukaryote. In that only the DNA replication takes place. Which of the following statements is not true about DNA replication? Not true, asking question. So what is the answer? These are numerous different bacterial chromosomes with replication occurring in each at the same time. That is the answer for me. Okay, our next question is very easy. You can say very easily. The first codon to be discovered was dash. Which codes part? The first codon. Nonsense codon also we can see that one. already the triplet codons, genetic code taken the class for you. In that one, they explain about the codons. Do you understand? Based on the amino acids, based on there is a codons, we can see the 64 kinds of amino acids in your book. They given in the chapter about the codon system. Okay, so the first codon to be deciphered was dash. Which one is the first codon? Can you say? U.
and phenylalanine.
Phenylalanine. The phenylalanine is one of the amino acid. So the first codon is one of the key question mark. The first codon is described, that is a UUU, the code word. Next, which codes are representing what type of amino acid? Phenylalanine. Next, Meslison and Stolz experiment proved dash. Proved dash. Whether a transduction or DNA is a genetic material or transformation or semi-conservative nature of the DNA replication. Which one is correct?
Semi-conservative nature of DNA replication.
I think everyone are marking in your book. Okay, mark it correctly. If you have any doubt, you can ask me in the next class. If you have any doubt in the one verse or any 2 mark, 3 mark exercise question, you can ask in the next class. Next one, see. 12th question. Ribosomes are composed of 2 subunits. Ribosomes are composed of 2 subunits. The smaller subunit and larger subunit. You already studied the protein synthesis. In the protein synthesis, they given the like this diagram. See, they given the like this one diagram. See in your book, they given that. This is a larger unit. This is a smaller unit. Okay, some like that number of the RNA are coming under the joint link, like that given in your book. Okay, so that's it. The ribosomes are composed of 2 subunits. The ribosomes are composed, this one ribosomes. Okay, the ribosomes are composed of 2 units. One is a smaller unit, another one is a larger unit. The smaller subunit of the ribosome has a binding site. It is one of the binding sites. See in your diagram. And the larger subunit has a 2 binding sites for 2 dash. So which one is which answer is coming there? First one, mRNA. First dash mRNA, messenger RNA. Next one, transfer RNA. Next one, transfer RNA. Messenger RNA and transfer RNA. Next one, an operon is a dash. Black operon model is explained in the last class now. So an operon is a dash. Can you say? An operon is a very important question. Mother, one black operon model. So the operon is a dash. It's a
Cluster of structural genes with related function.
Related function. Okay, next, last question. When lactose is present in the cultural medium. When lactose is present in the cultural medium. We are using the lactose for growing up purpose of the bacteria cells. The lactose is present in the cultural medium. Where we are using lactose, we already studied operon and lac operon model. In that one, we studied about the lac Y, Z, and A. Before page you see, that is the diagram given, see that. The given lac operon model, Y, Z, A, like that genes. Transcription of the lac Y, lactose, lac means lactose Y, lactose Z, lactose A gene occurs. As well as repressor is present. Repressor is unable to bind with the operator. Like that given in the diagram. So for this, the question answer is both A and B are correct. Transcription of the lac Y, lac Z, lac A gene occurs there. As well as the repressor is unable to bind the operator. So both the statement are also correct based on the lactose in the cultural. Do you understand? See, if you have any doubt, you ask me. Okay, or otherwise you call me, I will explain that one, or I will clarify your doubts. Okay, for the next week test, the 5th class will only one. So read properly. Okay. Can you hear my voice? Thank you, student. Thank you everyone. Whiteboard வேணுமா சார்? Whiteboard வேணுமா சார்? Whiteboard வேணுமா சார்? Whiteboard வந்துச்சு சார். கூப்பிடுங்க சார். Right.